lecture on protein misfolding, the clinical aspects of it. So we will start off by describing protein misfolding. This happens when, as the name suggests, protein is improperly folded, right? Now, if one protein is misfolded, that can easily be tagged and removed from the body. But if more than one protein are misfolded, then you might have these things called aggregates of proteins. So proteins will aggregate, and aggregated proteins are not easily gotten rid of from the body. And this causes problems, as we will see shortly. We will start by discussing Alzheimer's disease. Now, normally, on the diagram on the left is the normal condition. The diagram on the right is the abnormal or diseased condition. So normally you have this plasma membrane of the nerve cell, also called neurons, and you have a transmembranal protein called amyloid precursor protein, abbreviated as APP. Now, you have these two enzymes, one called alpha secretase, another called gamma secretase. Now these cleave or cut the APP at specific locations. So alpha secretase will cut or cleave the APP at this location, and gamma secretase will cleave the APP at this location. So the cleaved portion is gotten rid of by the body. Now, basically, I forgot to mention that APP has normal functions, right, in the nerve cell. It helps the nerve to grow and be nourished or something. But it has functions. It helps the neuron thrive. And this is all normal, right? You remove the cleaved portion of the APP once it's no longer needed. But things can go wrong. What sort of things? Well, instead of alpha secretase, you might have this enzyme called beta secretase. Notice how it cuts the APP at a different location from that of alpha secretase. And so you result in this thing over here. This cleaved portions the body cannot get rid of, and this cleaved portion is called amyloid beta. So the body cannot get rid of amyloid beta. And this will just stay outside the neuron. It will accumulate, because it's not going to happen only once, it's going to happen multiple times, and this thing will get sort of clogged between neurons. So you have here one beta amyloid. This is one beta amyloid. This is another beta amyloid. So you have many beta amyloids accumulating together. And this accumulation of many beta amyloids is called a beta amyloid plaque or plaque, right? I think it's called plaque. So you have many beta amyloid plaques, and they come in between neurons. You have many neurons, and in between those neurons, you will have these beta amyloid plaques. And this is problematic because if you have beta amyloid plaques interfering between neurons, then you will disrupt the normal communication between those two neurons. So imagine this is a neuron, you have another neuron somewhere here, and you have these beta amyloid plaques in between. You will disrupt the normal communication between this neuron and this neuron. And if you disrupt the communication between neurons, information is not going to be relayed from one neuron to the other. And this happens normally with neurons that are responsible for memory. And so let's say this neuron is responsible for memory, so is this neuron. If you insert these beta amyloid plaques in between these two neurons, memory is going to be impaired, which is one of the reasons why people with Alzheimer's, they sort of have dementia, right? They have memory disorders. Now this was outside the neuron, right? Beta amyloid plaques, they accumulate outside the neuron, in between neurons and on the outside, on the extracellular. But there's something else that happens with Alzheimer's, and it happens inside the neuron. Before we discuss what that thing is, let's first describe something normal that happens in neurons. So inside one neuron, you have this transportation network called a microtubule. Microtubule basically, as we said, is a transportation network. It transports nutrients, information from one part of the neuronal cell to another part of the neuronal cell. Now these microtubules, they need to be stabilized. They are stabilized by this protein called tau. You have many tau proteins and they stabilize the microtubule. And tau is a normal protein. It normally stabilizes the structure of the microtubule, thus allowing for proper, transportation network inside the neuron. But things can go wrong, like hyperphosphorylation of tau. Now, what does that mean? It basically means inserting many PO4s onto tau. You're hyperphosphorylating it. Phosphorylating really means just adding PO4, a phosphate group. Now, when that happens, tau loses its ability to function properly. When it loses its ability to function properly, it accumulates with many other tau proteins who have also been hyperphosphorylated improperly. And so if many tau proteins together, they form this thing called a neurofibrillary tangle, right? And this is something this is what neurofibrillary tangles look like. You have many tau proteins together. Now, what happens if tau proteins don't function normally? What happens if tau proteins, they accumulate and they become clogged inside the neuron cell? Remember we said tau was responsible for the stabilization of microtubules. If tau is not carrying out its function, the microtubule is not going to be stabilized. And so the transportation system inside the neuron will be disrupted. So nutrients from this part of the neuron, say, is not going to be transported to this part of the neuron because you have disruption in the transportation network. Why? Because tau protein has been hyperphosphorylated and it becomes clogged inside the neuron. It accumulates with many other tau proteins to form neurofibrillary tangle, which become clogged inside the neuron, right? Tau proteins, neurofibrillary tangles, they accumulate inside the neuron. Remember, beta amyloid plaques, they accumulate outside the neuron. Now, accumulation of neurofibrillary tangles can result in neuronal death, and neurons that are responsible for memory. And this is another reason why people with Alzheimer's will have memory problems. All right, prion diseases. Now again, normally in the plasma membrane of neurons, we have this protein called prion protein, and this is normal, right? All of us, we have this prion protein in us, and prion protein's function is, I think, up to this day unknown. But anyways, it exists and it is normal. And this is how a normal prion looks like. It is mainly made of alpha helices. Remember how we discussed that in secondary structure you have alpha helices, beta pleated sheets. Now prion proteins, they mainly have alpha helices, right? And this normal prion protein is what we call PRPC. What happens if it becomes infected? If this prion protein becomes infected, it will change to form this thing over here called PRPSC. Now prion protein SC has its alpha helices replaced with beta sheets, and this is problematic because it may cause problems, as we will see shortly. So you have this normal prion protein, and you have this abnormal prion protein over here. The normal one is called PRPC and is made of mainly alpha helices. The abnormal one is called PRPSC and is made of many beta sheets, right? Now, prions can cause a disease called transmissible spongiform encephalopathies, abbreviated as TSE. If this occurs in humans, we call it Creutzfeldt-Jakob disease. If it occurs in cows, we call it bovine spongiform encephalopathy, also known as mad cow disease. Now, these abnormal prion proteins, the PRPSC, might cause these disturbances over here, might cause memory disorders, insomnia, depression, thinking capability, depression, visual disturbances, and impaired judgment, and so on, and then etc. So there are many problems that are associated with abnormal prion proteins, which is PRPSC. Huntington's disease. Now, you have this thing called glutamine, right? It's an amino acid, and it is made up of CAG. You know, each amino acid has three. The codon of glutamine is CAG, right? Now, normally, we have less than 27 repeats of CAG. So in other words, CAG is repeated less than 27 times. This is normal, right? Now, in some people, CAG will be repeated 40 times. Now, if it is repeated 40 times, it causes something called Huntington's disease, which is basically a neurodegenerative genetic disorder. We call it a neurodegenerative because neuro from the brain, and degenerative because it occurs, it gradually degenerates the brain on the long term, right? So you start off with a few symptoms at the beginning, and then you gradually, right, exacerbate those symptoms. They become worse with time. Now, this disease will cause muscle coordination. It will affect the normal muscle coordination, and Huntington's disease also leads to cognitive decline and dementia. So basically, these multiple repeats, these excessive, 40 times repeats of CAG, it's called polyglutamine repeats. And as we said, it is a neurodegenerative disease, causes has negative effects on muscle coordination, and it might lead to cognitive decline and dementia. Notice here, this is a normal brain, and this is a brain of a person suffering from Huntington's disease. Notice how the brain is shrinking, and it's shrinking because neurons are dying. Why are they dying? Because of the polyglutamine repeats. Now, this polyglutamine is going to aggregate basically on the neurons and cause neuronal death, as you can see in this diagram. Parkinson's disease. Now, what you're looking at is a neuron. In this case, is a neuron in a region of the brain called the substantia nigra. Now, the substantia nigra primarily functions to control voluntary movement, right? So you have this neuron here and the substantia nigra. Inside this neuron, you have, normally all neurons in the substantia nigra will have this thing called alpha-synuclein, right? Whose function I'm not sure of, but you have alpha-synuclein in the neurons of the substantia nigra in the brain. Now, these alpha-synuclein proteins might accumulate and aggregate to form what we call fibrils, right? And many fibrils together will form this thing called Lewy Bodies. So Lewy Bodies is nothing more than the accumulation or the aggregation of alpha-synuclein proteins. Now, if you accumulate these alpha-synuclein proteins, you will form what we call Lewy Bodies. And Lewy Bodies have shown to cause neuronal death, as you can see over here. And so if you if you allow the neurons to die in the substantia nigra, you will lose control over your voluntary movement. And in the case of Parkinson, the voluntary movement that comes to mind is the movement of the hand. So neuronal death occurs at the substantia nigra because of the accumulation of Lewy Bodies that have many alpha-synuclein. And because of that neuronal death, people suffering from Parkinson's will have shaky hands, tremors we call them. Again, Parkinson's disease is your neurons in the substantia nigra primarily will have these intrusions in them called Lewy Bodies, which is made up of many alpha-synuclein. So we're done with the diseases of the brain. Let's go with sickle cell anemia. Now remember this diagram from the previous video? This is a hemoglobin structure. Remember hemoglobin has the ability to form quaternary structure, right? You have this tertiary structure over here, this tertiary structure over here, this tertiary structure over here, this tertiary structure over here at the back. Now each tertiary structure is called a chain. You have 4 chains, 2 of them, because you have 4 tertiary proteins, so you have four tertiary structures, so you have 4 chains. 2 of them are called beta chains and other 2 are called alpha chains, right? Now in the case of sickle cell anemia, alpha chains are unaffected, they're normal. The problem is with the beta chains of the hemoglobin. Now what really happens? A mutation occurs, a point mutation, which I think you are familiar with, right? So glutamine is replaced by what we call valine. Now, if you remember, valine is a hydrophobic amino acid, and I leave it to your imagination of what might happen if you have a hydrophobic amino acid in blood, because hemoglobin is in blood, right? Now, to give you a hint, this will be the structure of hemoglobin. Now, obviously, if you have this weird architecture of the hemoglobin, there will be disruption in normal hemoglobin functions, like carrying oxygen throughout the body. Cystic